Hi, my name is Cruz, and I'm a member of the Global Research Laboratory at Fresno State, and this is Counting Flies, exploring potential neuroprotectants for BMAA-induced neurodegeneration in Drosophila and melanogaster. Central to this research was BMAA, which is a naturally occurring non-protein amino acid that is produced by both symbiotic and free-living species outside of bacteria. The leading hypothesis for BMAA is that it is an environmental neurotoxin that is associated with Guamanian amyotrophic lateral Sclerosis-Parkinsonism dementia complex, which is almost exclusively an environmental disease with no link to genetic mutation. The support for this hypothesis comes from research showing that increased exposure through chronic dietary consumption of BMAA results in increased motor neuron loss, muscle atrophy, functional decline, and dementia in patients suffering from ALS-PDC. Some of the major concerns with BMAA is that it can be biomagnified, which can lead to the potential exposure in large concentrations. Special cases of the Guamanian ecosystem, where both the free form and the protein-associated forms of BMAA have been found to biomagnify from cyanobacteria up to cycads and flying foxes, which can then consequently deliver large doses of BMAA to the Chamorro people of Guam, who use both of them as food sources. Another concern is due to the ubiquitous nature of the cyanobacteria. Researchers have hypothesized that BMAA might actually be more prevalent in other food sources or other food chains than previously thought. One of the mechanisms of chronic toxicity of BMAA is through its misincorporation into proteins during peptide synthesis, which then leads to the misfolding of those proteins and eventually the aggregation of those misfolded proteins, which is the hallmark biochemical results of the ALS and Alzheimer's diseases. This misincorporation also provides a mechanism of slow release for BMAA that is concentrated in those misfolded proteins when they are broken down. Previous research has shown that under high concentrations of the amino acid L-serine, this misincorporation can actually be prevented. Another mechanism is a hypothesized mechanism of excitotoxicity under the presence of bicarbonate and physiological pH, such as in the blood buffer system. BMAA can actually be carbamylated into its carbamate adducts, and these carbamate adducts structurally resemble the excitatory amino acid glutamate, which allows them to bind to nerve receptors, such as the NMDA receptor, which can then lead to damage and death of nerve cells through overstimulation of those receptors. Another mechanism is through the activity of the carbamates as metal chelators. The carbamates can chelate or bind to nickel, but more importantly, copper 2 and zinc 2, both of which are important active sites of the copper-zinc superoxide dismutase enzyme, or the SOD1 enzyme, which is an antioxidant enzyme. The binding of the SOD1 enzyme can result in the decreased ability to remove reactive oxygen species and then lead to the potential increase of oxidative molecules that can cause damage to nerve cells. So the main objective of this experiment was to measure any changes in the viability of the fruit fly under the presence of BMAA and compound X. In other words, we wanted to know whether compound X would inhibit the side effects of BMAA neurodegeneration through the inhibition of either the excitotoxic mechanism, knowing that compound X is a possible allosteric inhibitor of the NMDA receptor, or through the inhibition of the metal chelating mechanism, by knowing that compound X can actually chelate BMAA and vice versa. So we set up 3 feeding experiments, the first one being a co-feeding with various combinations of compound X, BMAA, and L-serine, with water as a control, L-serine at 20 millimolar, compound X at 5 millimolar here and everywhere else, BMAA at 25 millimolar here and everywhere else as well, BMAA and compound X, BMAA and L-serine at either 20 or 25 millimolar, BMAA and L-serine at 20 millimolar and compound X. The next one being a co-feeding of BMAA in combination of compound X treatments at different concentrations, water once again as control, BMAA also at 25 millimolar here, and BMAA and compound X at 5, 2.5, or 1 millimolar concentration. The next step was our pre-feeding experiment, where fruit flies were treated with a pre-feeding of compound X for 5 days, and then transitioned to BMAA for another 5 days. BMAA still at 25 millimolars in this case, and our pre-feeding was with water. And in this case, we pre-fed the fruit flies with 5 millimolar, 2.5 millimolar, or 1 millimolar concentrations of compound X, and then transitioned to 5 days of BMAA. The treatments were administered in a food pellet of 500 microliters with an agar base, and 20% of that volume being the added treatment. We used 10 virgin female fruit flies of the Canton S strain per treatment type, with a 1 123 age difference in the fruit flies, and we incubated the fruit flies at 22 degrees Celsius with a 12 to 12 light dark cycle. We recorded the viability of the fruit flies in each of the treatments in 24-hour periods for 5 days. And for our first results of our first feeding experiment, with our treatment type on our x-axis and the average percent of fruit flies that were alive on our y-axis, we see that similarly to our control, our L-serine and our compound X sole treatments essentially had no effect on the fruit fly viability, but our BMAA treated groups have a sharp decline in the number of fruit flies that were alive by the end of the 5th day. And it's actually summarized up here. These are the average percentage of the fruit flies that were alive by the end of the 5th day. BMAA being the only significant number that's actually different from our control. Only 10% of the fruit flies were alive by the end of the 5th day. Our BMAA and our compound X at 5 millimolar, we see a 10% increase in the viability of the fruit flies by the end of the 5th day, but no significant difference between these two groups here. Our BMAA and our L-serine at 20 millimolar, we see a 10% viability in the fruit flies at the end of the 5th day. Our BMAA and our L-serine at 25 millimolar, we see a 5% increase from that to our 20 millimolar concentration, but once again, not a significant difference to our sole BMAA treatment. And our BMAA, L-serine, and compound X, a 5% increase than that to our BMAA sole treatment, but still not significant. Similarly, in our co-feeding treatments with BMAA and compound X, we see that our BMAA groups have a sharp decline in the number of fruit flies that were alive by the end of the 5th day. That's also summarized up here. Our BMAA group being significantly different than that of our control group, with 50% of the fruit flies alive by the end of the 5th day. That average increase of 10% also observed here in our BMAA and our compound X at 5 millimolar concentration. Our BMAA and our compound X at 2.5 millimolar, we actually see a decline to 3% of the fruit flies that are alive by the end of the 5th day. And our BMAA and our compound X 1 millimolar concentration, 17%, similar to that of our BMAA treatment group. Once again, none of these are significantly different to that than our BMAA sole feeding treatment. Lastly, for our pre-feeding treatment, what we saw was that characteristic sharp decline in the viability of fruit flies by the end of the 5th day. In this case, our BMAA 25 millimolar is still significant, 23% by the end of the 5th day. And our compound X in this case at 5 millimolar, we see not a big difference to that, to our sole BMAA treatment, with actually a lower percentage. Our compound X 2.5 millimolar treatment, we see that it's actually lower as well. And in this case, our compound X and 1 millimolar concentration, we see that average 10% increase in this treatment, but still not a significant difference between them. And I just want to leave off with physical representation of the effects of BMAA in these flies. So this is characteristic of all treatment BMAA treated flies, where they have the inability to walk and they have muscle sporadicity. So going back to our main question, whether the consumption of compound X would increase the viability of the fruit flies by alleviating any of the side effects of BMAA-induced neurodegeneration. Well, we saw an average increase in 10% in the viability of the fruit flies by the end of the 5th day between our BMAA and our BMAA plus compound X treated flies, but that difference was actually not very significant. We also didn't see that alleviation of the BMAA-induced neurodegeneration under the concentrations of L-serine, even at equimolar concentrations. And however, we do show support for the neurodegeneration of BMAA through the chronic consumption mechanism. One of the mechanisms is possibly the excitotoxic mechanism due to the muscle sporadicity and inability of the fruit flies to walk around. Thank you.